Hello students, welcome to Zoology Classroom. In today's lecture, we are going to discuss about RNA structure and its types. So, DNA is the genetic material, okay, we know it. So some parts of the DNA makes the genes, and these genes, they will produce proteins, and these proteins, they will control the traits in our body. So in the process of converting information which is present in the DNA into proteins, some special type of nucleic acids are required. They are called RNA. So RNA is very, very essential in converting the information from genes to proteins. So RNA, like DNA, it is also a polymer of nucleotides. But here the nucleotides present in RNA are called as ribonucleotides. And all these ribonucleotides, they are linked to each other by 3' to 5' phosphodiester bonding. So, and each ribonucleotide is made up of 3 moieties or 3 components. One is phosphate, that comes from phosphoric acid. Second one is ribose sugar, and third one is nitrogenous base. So here in nitrogenous base, we have 4 types of nitrogenous bases in RNA. Okay, so out of them, 2 are purines and 2 are pyrimidines. In purines, we have cytosine and uracil. Uracil is present in the RNA in place of thymine. Thymine is present in DNA but not present in RNA, and uracil is present in RNA but not in DNA. So the cytosine and uracil, these are single ring structures with hexagonal rings. With the hexagonal structures, they are pyrimidines. And purines are 2 ringed structures. One is a hexagonal ring and one is pentagonal ring. Adenine and guanine are the purines. So 4 types of nitrogenous bases are present. So out of these 3 moieties, pentose sugar is attached to nitrogenous base with its first carbon, and pentose sugar attaches to phosphate group by its 5th carbon. So totally 4 nucleotides are there. So we can see 4 types of nucleotides. 4 nitrogenous bases are there. So we can see 4 nucleotides. So one is having adenine, another one is having uracil, and third one is cytosine, and fourth one is guanine. So these are the 4 nucleotides that we can see in RNA. So all these nucleotides, they are attached to each other by 3' to 5' phosphodiester bondings. 3' to 5' phosphodiester bondings unite all these individual ribonucleotides with each other, so that finally a polynucleotide is formed. And these RNA, usually these RNAs are having some differences when compared to DNA. So let us see what are those differences. And usually RNA is single stranded, DNA is usually double stranded. But in rare cases, in RNA, at some places we can find a double stranded structure. These will form some hairpin-like structures or loops in RNA, in some cases only. And ribose is the main sugar part in RNA. Deoxyribose is the sugar part in DNA. So in the pyrimidine component is, one pyrimidine component will be different in RNA and DNA. So thymine will be present in RNA. In place of thymine, thymine is absent in RNA. In place of thymine, uracil is present in RNA. So in place of uracil, we will see thymine in DNA. So Chargaff rule is there. In this Chargaff rule, we know that complementary base pairings will be there. Adenine always binds with thymine and guanine always binds with uracil. So as RNA is single stranded, no Chargaff rule cannot be seen in RNA except in some cases where we can find double stranded parts in the RNA. And DNA, as it is double helical structure, so two polynucleotide chains are there. So in forming nitrogenous base pairs, Chargaff's rule is followed in DNA. And RNA can easily destroyed by alkaline materials, but DNA is not destroyed, easily destroyed by alkaline material. Why? Because it is lacking hydroxyl group in it, in the second position of the pentose sugar or deoxyribose. And RNA is relatively labile molecule. That means it is easily degradable, but DNA is a stable molecule. It will not be degraded easily. And RNA, mostly it is present in cytoplasm. Only some amount of, some type of RNA are present in nucleus, but DNA is mostly found in nucleus. In some cases, DNA can be seen in extranuclear places like in mitochondria or in chloroplast. The base content or nitrogenous bases number is limited in RNA. Mostly 1000 to 5000 nitrogenous bases or nucleotides are seen in RNA, but in DNA, millions of nucleotides are present in DNA. And coming to different types of RNA or DNA, DNA is single type. No different types of DNA are present in any type of organism. But in RNA, several types of RNA are present, like mRNA, messenger RNA, rRNA, ribosomal RNA, tRNA, transfer RNA, snRNA, siRNA, miRNA, hnRNA. Like this, several types of RNA are present in almost all types of cells. And all these RNA types, they always possess they always possess the same form, so their forms will not be changed. But whereas coming to DNA, DNA will be having three different types of forms: aDNA, bDNA, and zDNA. Depending on the internal conditions, DNA can change its form from A to B form, B to Z form, like that. So and RNA is synthesized from DNA, and usually DNA cannot be formed from RNA, but in some cases, usually in some viruses, reverse transcriptase enzyme will be there. This reverse transcriptase enzyme is capable of producing DNA from RNA in reverse manner. And coming to DNA, DNA can form RNA, and DNA also can form DNA. And many copies of RNA are present in each cell, but only single copy of DNA will be present in each cell. So each cell possesses only one copy of DNA, but not many copies like RNA. And coming to types of RNA, so in all prokaryotic and eukaryotic organisms, we can see three major classes of RNA molecules, like messenger RNA, mRNA, transfer RNA, tRNA, ribosomal RNA, or RNA. Apart from these major classes of RNA, some other minor classes are also present. They are small nuclear RNA, snRNA, microRNA, miRNA, and small interfering RNA, siRNA, and heterogeneous nuclear RNA, hnRNA. Let us see the details of these types of RNA one by one. Coming to first one is messenger RNA. This is also called as mRNA. This is very, very important one. It comprises of 5% of total RNA present in the cell, and it is coding RNA. So out of remaining all RNAs are non-coding RNAs, but this mRNA is called as coding RNA. Why? Because it possesses codons. Codons are very, very important signals required for the production of protein synthesis. So these signals required for protein synthesis are present in mRNA. So that's why it is called as messenger RNA. Why? Because it is carrying the message of the DNA and it is carrying that message to the area where protein synthesis is being done. So, and it is most heterogeneous in size and in sequence of nitrogenous bases also. So size is different from one mRNA to another mRNA, and nitrogenous bases sequence are also will be different. Whereas in case of remaining RNAs, size is limited, and nitrogenous bases sequence are also will be stable in remaining types of RNAs. And in the production of this mRNA, usually in the first case, usually in eukaryotes, heterogeneous RNA will be made, and this heterogeneous RNA will possess some unwanted or unrequired or not required parts. They are called introns. So in all those things will be deleted or sliced. That process is called RNA slicing. After RNA slicing is done, the finally mRNA will be produced. So this final mRNA also again it undergoes some changes. So in these changes, at the 5' end, one cap will be added. At the 5' end, one cap will be added. This cap usually will be the same in all types of mRNA, that is methyl guanosine triphosphate. So methyl guanosine triphosphate molecule will be added at the 5' end of the mRNA. And at the 3' end of each mRNA, poly A tail. So many nucleotides which are having adenine in them. So these adenylate nucleotides, many adenylate nucleotides will be added at the 3' end. So this cap, 5' cap, or this methyl cap is helpful to the mRNA while transcription as while translation is going on near the ribosomes. In what way it is helpful to the mRNA? So mRNA can be recognized by the help of this uh, methyl guanosine triphosphate capping only. So mRNA will be recognized by this cap. So this capping is useful for recognition of mRNA while protein synthesis is being initiated. And the 3' end is having poly A tail, and this 5' end is having this methyl cap. So this methyl cap and poly A tail, both these endings, both these terminal additions help to the mRNA in having stability also. So mRNA stability also will be assured by this 5' cap and poly A tail. So, and each mRNA will be having some portion of the mRNA will be having coding sequence. Here in this diagram, we can see this yellow color part is having, it is initiated, or it is having a beginning with AUG code, and this ending is having UAG code. So UAG is ending codon or nonsense codon. AUG is the initiation codon. So the region which is starting from initiation codon and ending with any one of the 3 nonsense codons is called coding sequence. So, and before this coding sequence and after this coding sequence also, some nitrogenous bases or some regions will be there, but these regions are not transcribed or translated into protein. So the region which is present before the coding sequence and in between 5' capping and coding sequence is called leader sequence, and the region which is present in between coding sequence and poly A tail is called trailer sequence. So these trailer sequences and leader sequences will not be translated into protein. This is about mRNA, and another one is heterogeneous nuclear RNA, hnRNA, as I have already told, in mammals and in eukaryotic organisms. So this DNA will be transcribed into hnRNA. So hnRNA is nothing but primary RNA. So here in this diagram, we can see blue parts and so the somewhat orange color parts. Okay, so these blue parts are called introns. They are not having required codon regions. These are unnecessary parts. And blue color ones are called introns. Blue color ones are called introns. They are having unnecessary parts. And these orange color ones are having required nucleotides for this protein synthesis. These parts are called exons. So this primary RNA or heterogeneous nuclear RNA or hnRNA it will undergo RNA slicing, a process called RNA slicing. In RNA slicing, all these introns and extrons will be sliced or cut. So after cutting is done, after slicing is done, unrequired parts or unnecessary parts will be removed and remaining parts will be added with each other in the same sequence. So after joining all these things, finally what happens before this slicing and removing of introns is being done, at the same time adding of methyl group to this 5' end and polya tail to the 3' end also will be done. So addition of polya tail, addition of 5-methyl endings will be accomplished while this removal of introns is being done. So finally, all introns will be removed and we will get mRNA. So hnRNA will be present only in the nucleus. After the hnRNA undergoes RNA splicing process, it will be converted into mRNA, and this mRNA will be entering into, it will be sent into the cytoplasm for translation to occur. So coming to this hnRNA, its molecular weight will be around 10 to the power of 7, and molecular weight of mRNA will be around 2 into 10 to the power of 6. So most part of the this what the hnRNA will be removed and finally it will be converted into mRNA. So after the all hnRNAs are primary RNAs, 75% of RNA material, hnRNA material will be degraded in the nucleus. Only 25% of its content will be processed into mature mRNA, and this mature mRNA will be exported into cytoplasm for translation to occur. And coming to another second type, tRNA, transfer RNA. So this is the smallest of three major types of RNA. So at least 20 types of mRNA, tRNA will be present in the cytoplasm. Why? Because there are 20 types of amino acids are there. Each amino acid will be having a special type of tRNA for itself. So, and each tRNA will be having 74 to 95 nucleotides in it, and it will be synthesized in the nucleus itself, and a precursor molecule will be produced in the nucleus, and after processing is done, it will be converted into transfer RNA or tRNA. This tRNA finally enters into cytoplasm, and these tRNAs, they will bring amino acids to the area where protein synthesis is being done. So they are soluble, so that they are called as soluble RNA or sRNA also. And they are also called adapter molecules. Why? Because during the protein synthesis is being done, they will bring amino acids and they will adapt to this protein synthesizing machinery, and they will transfer amino acids to the synthesizing protein chain. So that's why they are also called adapter molecules. And this tRNA, in the precursor form, it will be having primary structure. That means it will be having a straight chain like structure. But after processing, it will take secondary structure, and this secondary structure looks like a clover leaf model. So here in this diagram, we can see this is the clover leaf model of tRNA. This is the secondary structure of tRNA. This secondary structure contains 5 parts in it. One is acceptor arm. So here in this diagram, we can see that 3 end and 5 end. So we can see that at some places it is having nitrogenous base pairing. So complementary nitrogenous bases, they are forming pairings. So these pairings are giving this type of structure, clover leaf structure to this tRNA, that is secondary structure of tRNA. So, like any other nucleotide, nucleic acid, it also will be having 3' end and 5' end. So, in 3' end will be having CCA ending. So, 3' end will end with three particular nucleotides. They are having C, C, and A. Cytosine, cytosine, and adenine will be there. So in each tRNA, 3' end will be having this CCA ending. So this is called as acceptor arm. So this acceptor arm is attached to the amino acid. So amino acids are attached to the tRNA here at this 3' end to the CCA ending. So this is the first part, acceptor arm. So this is called acceptor arm. And second one is anticodon arm. So the acceptor arm, so this is in brinjal color. So opposite to this acceptor arm is called anticodon arm. So this is anticodon arm. Blue color one is anticodon arm. So this anticodon arm is having 5 base pairings in it. 5 base pairings are there in it. And this acceptor arm is having 7 base pairings. 1, 2, 3, 4, 5, 6, 7. 7 base pairings will be there in this acceptor arm. And in the anticodon arm, 5 base pairings will be there, and 5 individual nitrogen nucleotides will be there. So out of these 5 nucleotides, these three things are called anticodons, and these anticodons are complementary to the codons of the mRNA. So mRNA is linked to this tRNA here at this anticodon region. So as this arm is possessing anticodon, this is called as anticodon arm. And another one is D arm. So D arm will be on the 5' end side. So 5' end side, D arm will be there, and opposite to this thing, that means on the 3' end, we will be having T arm, or T pseudouridine C. T pseudouridine C. So in place of uracil, pseudouridine will be there. Pseudouridine is similar to uracil. So TUC arm will be there on 3' end. And fifth one is extra arm. Extra arm will be in between this anticodon arm and T pseudo uracil and cytosine arm, or TUC arm, or T arm. So in between T arm and anticodon arm, extra arm will be there, and this extra arm is variable in each type of RNA. In all RNAs, it is not same. So number of nucleotides present in this extra arm will be changing from one tRNA to another tRNA. Remaining all parts will be having the same amount of nitrogenous base pairing, same amount of nucleotides in them. For example, this T arm will be having 5 nitrogenous base pairings, D arm will be having 4 nitrogenous base pairings, anticodon arm will be having 5 nitrogenous base pairings, and acceptor arm will be having 7 nitrogenous base pairings. So these base pairings, the number of free nucleotides, all these are same in all 20 different tRNA molecules. What will be changing in 20 different tRNA molecules is this extra arm only. So this is about secondary structure of tRNA. And in some cases, the secondary structure takes tertiary form also. So this both these two arms, D arm and T arm, they will take some turns, and they will form some bondings in between them. So finally, the cloverleaf model will be converted into this tertiary structure. So usually, this tertiary structure tRNA molecules are also present in the cytoplasm. Coming to third type of major types of RNA, that is ribosomal RNA or RNA. So rRNAs are very small chained these chains are these ribosomal RNAs are very important in the formation of ribosomes. So several types of ribosomal RNAs are present, and in prokaryotes 23S rRNA, 5S rRNA, and 16S rRNA. So three types of rRNAs are present in prokaryotes. Out of these 23S and 5S types, both these two things along with 31 ribonucleoproteins, they will form major subunit, that is 50S subunit. And 16S rRNA along with 21 types of ribonucleoproteins, it will make 30S subunit. And these two subunits, while the protein synthesis is going on, they will unite to form the 70S type of ribosome. Coming to eukaryotes, in eukaryotes 20S type, 5' type, and 18S type, and 5S. Okay, 28S, 5' and 5S and 18S. So totally four types of rRNAs are present in eukaryotes. Out of these four, 28S and 5' and 5S. These three, along with 50 different types of ribonucleoproteins, will form major subunit of RNA ribosome. And 18S rRNA along with 33 different types of ribonucleoproteins, RNPs, will make 40S type of subunit or small subunit of ribosome. So these two subunits, while the protein synthesis is being initiated, then they will unite to form 80S type of ribosome. So the function of these RNA molecules or RNA molecules is not known, is not fully understood, but scientists are believing that they are important for the assembly of ribosomes, and they also help in binding of mRNA to the ribosomes. And coming to the synthesis of this rRNA, so except 5S RNA, remaining all RNAs, they will be produced from a same precursor, that is called 45S precursor. So from this 45S precursor, remaining four types of RNAs will be formed. 5S RNA will be produced individually or separately. And recent studies saying that this rRNA components, they will help in forming this peptidyl transferase enzyme activity. So they will help in the peptidyl transferase activity, that in that way they are forming they are performing an enzyme role, so that along with ribosomes, these are RNAs performing a enzyme activity, so that's why they are called ribosome also. And apart from these three major RNAs, several small RNAs are also present in the cells. So they are present in the range of the range and the size or length of these small RNAs varies from 20 to 300 nucleotides, and many copies ranging from 1 lakh to 10 lakh copies are present in each cell. So they are present both in the cytoplasm and nucleoplasm. Let us see what are those small RNAs. Small RNAs. So in the small RNAs, first one is small nuclear RNA, snRNA. So this snRNAs, they are very important in RNA processing. So earlier we have already discussed that pre-mRNA is also called as heterogeneous RNA. So in this unwanted or nonsense regions or unrequired regions are present in the form of introns. So these introns will be sliced or separated, and the exons will be added to each other finally to make mature mRNA. So in this process, spliceosome enzyme will be useful. So this spliceosome is formed with the help of snRNAs. So snRNAs and some nuclear proteins or ribonuclear proteins, So small nuclear RNAs and small nuclear ribonucleoproteins, snRNPs, all these things. So snRNA and some special proteins form join together to form spliceosome, and this spliceosome removes these unnecessary intron parts. So in this way, small nuclear RNAs are very important in RNA splicing process, and they are also useful in transferring poly A tail to the 3' end of the mature RNA. And another one is microRNA. It is also called as miRNA, and third type is small interfering RNA, siRNA. So these two classes of small RNAs are present in the nuclei, and they are helpful in inhibiting the gene expression by decreasing the specific protein production. So different types of mechanisms are there. So by undergoing these different types of distinct mechanisms, these miRNAs and siRNAs, they will inhibit expression of certain genes. In that way, they will decrease the production of some special proteins. In that way, they are inhibiting the protein production of some genes. So, and let us see one by one of these two RNAs, microRNA, miRNA, first one. So these miRNAs are typically, they will be having only small chains. So in them, they will be having 21 to 25 nucleotides only. So, and they process the products of certain genes. And these miRNAs, okay, these miRNAs, what they will do? They will react with this mRNA. So they will react with mRNA, and they will, what they will do, they will delete the, or they will destruct the RNA. So first they will form a bonding with this mRNA. So RNA and RNA duplexes will be formed. So in this RNA and RNA duplexes, one is mRNA and another one is this microRNA. So later this microRNA, it splices the mRNA. In that way, it sees that protein synthesis is not done. So here we can see this is DNA, this is mRNA, and this mRNA will be entering into translation process. Finally, it will give protein product, final product. So here in this protein synthesis process, first one is transcription, this is translation. So this microRNA, they will interfere in this translation process. So they are acting as regulators in this translation process, so that they will destruct this mRNA, and they will see that protein is not synthesized. So in some cases, some protein synthesis is not required to the cell. In such cases, microRNA comes into picture, and if by mistake the mRNA is produced, it will destroy that mRNA, it will disturb this translation process, and it will see that the protein is not synthesized in the cell. So in this way, microRNA are very useful to the cell, where by mistake in some cases mRNAs are produced, but the product of that mRNA is not required by the cell at that particular time. So whenever it is not required, in such cases, this microRNA, they will come into picture and they will disturb this protein synthesis process. So, and another one is small interfering RNAs, siRNAs. So the length of these siRNAs will be 21 to 25 nucleotides. But unlike the previous one, it will be having double stranded structure. So, and in the beginning, it will be synthesized from a big molecule, big double stranded RNA. So by undergoing splicing, many small sized final siRNA molecules will be produced. So, and these siRNA molecules like miRNA, they will form RNA-RNA hybrids with the mRNA. So, and this mRNA synthesis again will be disturbed by this siRNA like miRNA only. So here in this way, this is a double-stranded RNA This double-stranded RNA will be sliced into several small-sized siRNAs, and these siRNAs, they will join with some special proteins. These oval-shaped things are proteins, special proteins. So along with the special proteins, this siRNA, they will form spliceosomes, and these spliceosomes, they will join with this mRNA and they will cleave the miRNA. So this cleaved mRNA will not be useful in protein translation process in protein synthesis. So in this way, siRNA also, they will disrupt the protein synthesis process like microRNAs or miRNAs. So what is the significance of this miRNA and siRNA? Both these mi and siRNAs are present in the nuclei, and they will come into the cytoplasm also. So these things, they are useful to the scientists in producing some specific drugs. So in in targeting some special drug molecules, so this miRNAs and siRNAs will be useful. So in this way, scientists are trying to utilize this miRNA's quality and siRNA's quality or character in treating some diseases and in interacting with some drugs in our bodies. And in addition to that, in preparing some knockdown models or knockout gene models, also this siRNAs will be utilized. So in knockdown animals, a special type of gene will be deleted from the organism. So we know about this genetically modified organisms. In genetically modified organisms, what will be done? In place of one DNA or one gene, a new gene from another organism will be inserted into the genome. So the genetically modified organisms will be having a foreign gene, usually which is not present in the species. And these knockdown animals or knockdown organisms are also one type of genetically modified organisms. But in these organisms, one gene will be deleted from the genome. So in remaining genetically modified organisms, one new gene will be added from other organisms. In knockdown animals, one gene will be deleted from the genome. So scientists usually produce knockdown animals to study the effect of a gene. So they want to study the effect of gene if that gene is not present. So in the absence of a particular gene, what will be happening? They want to know. So to study that thing, they will produce knockout animals. So by studying the knockout animals, they will come to know what is the effect of if the gene is absent in the organism. If we know the effect of the gene when it is not present, then indirectly we can know the effect of that gene also. So in this way, to understand the effect of some genes, knockdown animals are very, very handy. So in producing knockdown animals are also these miRNAs and siRNAs can be utilized. So scientists are trying to use these miRNAs and siRNAs in producing knockout genes and in producing special therapeutic drugs which are useful for humankind. And finally, let us see what are the different types of RNA and what are their uses. So mRNA, as we know, it is messenger RNA. It brings information message from the DNA, which is required for protein synthesis, and it is regulatory in function. And rRNA, it is helping in translation process. It is helping in formation of RNA, and as recently discovered, it is also performing catalytic action or enzymatic action also. And tRNA, transfer RNA, they are also useful in translation process, and they are acting as adapters. hnRNA, and it is a precursor of mRNA and other RNA. Okay, other RNAs also will be primarily they will be called as hnRNA. After slicing is done, finally we call them as rRNA or tRNA like that. So mRNA, rRNA and tRNA, all these three RNAs will be initially produced in the form of hnRNA. After slicing is done, we call them as mRNA or RNA and tRNA like that. So this is the precursor RNA. And uh, small cytoplasmic RNA. The small cytoplasmic RNAs are also present in the cytoplasm, and they will be utilizing in tRNA processing, and they are also having catalytic function or enzymatic function. And snRNA and snORNA is another one, small nucleolar RNA. So the small nucleolar RNA also will be present in the nucleus. Both these two things will be helpful in mRNA processing, adding poly A to the mRNA in the 3' end, and rRNA processing, maturation and methylation. In all these activities, these two things are useful. And this siRNA and miRNA, they are regulating the translation process by disturbing the translation activity. So these are the different types of RNAs and their functions. So with this, today's topic is over. Let us meet in next class with another topic.